We said that in the spinal cord we can divide it into gray and white matter. For some reason they say that the gray matter is divided into horns and the white matter is divided into columns. Okay? What you want to do when you look at one of these things is you want to orient yourself. You want to know which way is towards the back of the body and which way is towards the front. There's a couple different ways to tell. One is that you see how one side of the butterfly touches the edge and the other side doesn't. The side that touches the edge is always posterior. So this is always the back. The other way is that if I tilt it up, you can see how there are these big bulges on one side, and see how that big bulge ends up hooking to this part of the spine. This is it's called the dorsal root ganglion. You will all learn to recognize it. And this is another way to see that this is the back side, the side with that hooks up to the bulge. Okay, so this is our back. Once we can identify which is the back and which is the front, it's easy breezy. So we look at the gray horn and we say, there's always three. There's a posterior, a lateral, and an anterior. So there's the posterior gray horn, the lateral gray horn, and the anterior gray horn. You'll never get them wrong as long as you know which side is towards the back and which side is towards the front. We can use that exact same terminology for the columns. So we have posterior white columns, lateral white columns, and anterior white columns. Like I said, we need to know the central canal, which is the hole in the middle, right in the center, through which the cerebrospinal fluid flows. That's all this proper. Now what we need to do is figure out all the roots and nerves that come off of it. So what happens is here's your central nervous system, right? And eventually goes all the way up to the brain, right? Well, coming in from the body is all the messages from your senses, and leaving the spinal cord are all the messages going to your muscles. So we have things sticking off of it. All the messages coming into your body come in through the back. So they're going to come in. All the cell bodies are going to sit in these bulges called the dorsal root ganglia. Each one individually is a dorsal root ganglion. So here it is. It looks like a spring onion, a lot of people will say.
Which one?
This one?
Okay.
Or number 19. Okay? So these are the dorsal root ganglion. That means that this one here and this portion here is considered the dorsal root. In other words, this is where the spinal nerve roots into the spinal cord, and it's the back part, so it's the dorsal root. And this is its bundle of cell bodies, so that's the dorsal root ganglion. Now we have a ventral root. You can see it in yellow here coming in, and it hooks up to the front side of the spinal cord. This is the ventral root, okay? This is going to be carrying information out to the muscles. So sensory comes in the back, motor comes out the front. Once they join together, so we have the two roots, they join together. Now the information is all mixed. We've got motor traveling and spinal and sensory information traveling together. That makes up a spinal nerve, which we just looked at. So when we saw all those different neurons, right, inside that bundle, some of them are sensory and some of them are motor. They're sending messages and receiving messages. So you can see that eventually the ventral and the dorsal root join together here. So this portion would be considered the spinal nerve. Now, this isn't on your sheet, but let me just point out to you, what's then going to happen is that that nerve branches. See how it's branching? So we have a dorsal branch and a ventral branch. So what's going to actually happen is that now this contains, again, motor and sensory information. This dorsal branch is going to go around and talk to the back of the body. So it's going to pick up sensory information from the back, and it's going to send out motor commands to the back. This much larger root, the ventral root, is going to do the opposite. It's going to come around the front of the body and speak to all the muscles in the front in that area, and it's going to pick up sensory reception from all of the organs in that area. Okay? So quite literally, you are divided by the spinal nerves. So this time is talked to by one spinal nerve, and then the next, and then the next, and then the next, and the next, because each one comes out and talks to a particular area. So we'll see a picture of that in just a little bit. Now, the other thing that we want to do, somewhere in here, you need to learn about the meninges. What the meninges are, and they're under the brain at the very end, is a series of protection for your central nervous system. It is present in the brain. We do not have a brain model that has the meninges on it. So we're going to do that on this model instead. You have 3 layers of meninges. You have the dura mater, which is the most outside. You have the arachnoid layer, which is in the middle, and then the pia mater, which is actually attached to the central nervous system. So in our model, G here, this is the dura mater. Okay? In the spinal cord, it sits away from the bone. And instead, this space outside of it is filled with fat. That's the epidural space. So when somebody gets an epidural, they're shooting pain medication into this fatty layer. Anytime you shoot into fat, which is very common because we do sub-Q shots all the time, it slows the rate of the dispersal of the medication and gives the patient a more long-lasting, steady rate of delivery of the medication. So we shoot it into the subdural space. You can see epidural space part of which you can see is highly vascularized. We're going to get that pain medication diffusing out to the patient over a period of time at a very steady rate without ever getting close to the spinal cord itself. It's a very effective way of going ahead and blocking sensation. So this is the epidural space. G is the dura. You can see here the next layer is very, very close. This is the arachnoid layer. So in between them, you can see between this layer and between this layer, they've put a little layer of peach in there. There is actually a layer, a space in there called the subdural space. The subdural space is an area, by contrast, which should not be filled with anything. So when somebody has a subdural hematoma, that means blood has accumulated in this spot, forced this layer down towards the brain, and is most likely now putting significant pressure on the brain, which is bad. So we don't usually have fluid in that space. This is the arachnoid, so the layer right below it is called the subarachnoid space. This, by contrast, is nice and open, just like we're seeing in this picture, because this is where the cerebrospinal fluid sits to bathe the spinal cord. The final layer is the pia mater. This is the white layer that's physically attached to the spinal cord. This is like a sponge, so it's going to like sponge up all that cerebrospinal fluid and keep it right next to the spinal cord itself, because that's where the spinal cord gets all its glucose and oxygen from. Okay? Okay. So we did all that. Let's move to the brain. Alright. How much pages of crap?
Yep. Yeah, like 2.
She said that this one is further, that's the back side. That's
Okay. So these brain models are kind of delicate. I hope it doesn't fall apart as we're doing this. The first thing that you want to do is just the big stuff. All of this part that's got in this model covered in the blood vessels, it's all foldy and peachy like this, all of this is cerebrum. In us, that's huge because we are very cognitive thinking people, right? We've got these huge cerebrums. The cerebrum is actually divided in half along the length of the brain. So this would be like the front of your forehead all the way to the back. It's divided into 2 halves, which we call hemispheres. So we have a right hemisphere and a left hemisphere. And we'll talk about in class how function is different. Some function is right and some function is left. Now you can see that the brain is folded. Why is the brain folded?
more space.
Surface area, more space. It's one of the major things that separates us from birds and lower animals. We have really folded brains. Reptiles, nice, smooth brain. Okay, they don't have as much cognitive ability. Each of the foldings is called a gyrus or a gyrus. People say a bunch of different names, and a bunch of them are called gyri. Each depression is called a sulcus or a sulci, or a sulci, I've heard people say too. Okay. Now, that fissure, like I was saying, that divides the hemispheres into the right and left, that goes from the front of your head all the way to the back, is called the longitudinal fissure. And it really is a fissure, meaning it runs deep into the brain. So if you've got somebody's real brain, you could easily open it up at the 2 hemispheres and pull and look inside. When you get your sheep brain, you'll see that once you cut through the meninges, you could again squeeze open the brain along the longitudinal fissure. sulcus is this little division here that divides the temporal lobe near the ear from the parietal lobes up where the parietal bone should be in the head. The central sulcus, which is this bad boy here, which is not that well defined on these, but is a little bit better on the right hemisphere. This central sulcus is going to separate the frontal lobe from the parietal lobes. Okay? So the division is not exactly where the bones are. So you want to note that as you begin to study. So this central sulcus is usually further back than the frontal bone would go. So we have a number of different lobes. We have the frontal lobe, which extends to the central sulcus, then two paired parietal lobes that extend from the central sulcus back. At the back of the brain, there's another sulcus. You guys don't have to learn the name of it. It's called the parieto-occipital sulcus. But it kind of comes up just like that lambdoidal suture on the back and forms a triangle. So this area back here is the occipital lobe. And then as we said, here to the side, separated by this lateral sulcus, we have the temporal lobe. Now, let's flip the brain over. So now we're going to go ahead and flip the brain over and start looking at some things underneath the brain before we bother to even open it up. Okay, so we'll come back to corpus callosum, I promise you. But in the meantime, so we're going to skip ahead. You have coming off of your brain 12 cranial nerves. And you guys are going to learn to name them all and know their functions and all those awesome things. A lot of these next few ones are in your handout are referring to the cranial nerves. This one, 28, are called the olfactory bulb. So what sense is it? Smell. So this is what comes down and sits right on top of your nose. And these little bulbs are going to send out projections, the end of which are going to be sensitive to chemicals in the air around you. So as you breathe in, all the chemicals bind to those hairs and they cause you to say it smells bad in here, it smells like a dead cat, whatever it is that you're smelling, okay? That's the bulb portion. It's then got to take that information and send it back to the brain to be processed. The only thing that's happened is it's caused electrons to send a neurons to send a message. We don't know what's happening yet until it hits the brain. The place where it starts to send the message back to the brain, this is the tract. So the bulb is at the end where we receive the message. The tract is sending that information back to the lobe where it will be processed. Then we have the optic system. In the optic system, you have what's called the optic nerve. You don't actually have that in the olfactory bulb because there's not anything beyond this. Basically, the olfactory bulb is not really an optic nerve, even though you will learn it as, it's not a nerve, cranial nerve, even though you'll learn it as such. The optic nerve truly is a nerve, so it's going to come out, it's nice and mixed, and head to each of your little eyeballs that would sit here, right? The information in your eyes crosses. So as each eye sees the whole world, what's going to happen is that the inside portion of that is going to cross sides, so that by the time it gets back to your head, the right half of your brain sees the left half of the visual world, and the left half of your brain sees the right half of the visual world. In order to do that, we need to cross some information over. That's called where it crosses the optic chiasm. And you can see in here that the information really does cross. So you can see that it looks like it forms a cross, which is how it got its name. Once it's crossed, and now it's sending the information on a spinal pathway back to the brain, deep inside here are the tracts. So that's the optic tract. So you want to learn it as three parts. Optic nerve goes towards the eyeball, chiasm where it crosses, tract taking it back to the brain. Now we're gonna skip a few things and we're gonna move on to one part that you can easily see, which is the cerebellum. This is the cerebellum. It's like it's a little separate brain underneath the brain. So when the brain is in your body, it's sitting on the back underneath. So you can see it sticking off there. That's the cerebellum. Okay, now we'll go ahead and we'll look inside. The reason that we have these models like this is so that you can see all the interior structures. The downside to them is that they fall apart really. So I'm going to lose the cerebellum and show you the rest of the brain. So here is my half of a brain. So let's go go back and pick up the corpus callosum as our first term. This is mostly gray matter. So in order to actually connect the two halves of the brain and to send information along, we have to have bundles of axons, the white matter. Part of that is here in the corpus callosum. So this is matter sends messages from one half of the brain to the other. And back in the olden days, this is what and actually they're doing it again, what they'll sever in epileptic patients. In epileptic patients, when they have a grand mal seizure, it means both hemispheres, the right and the left, are seizing. So how can you stop that? You can sever the corpus callosum. At that point, the right and the left hemisphere are operating independently of each other. This is our only point of contact between the two. Okay? So this is the corpus callosum. Now it's bigger than this. It extends into the brain, but we don't have a model here to show you that. Inside the brain, you actually have spaces. And those spaces are filled with cerebrospinal fluid. That's again what brings the oxygen and brings the glucose. The system of holes is called the ventricles. The first ventricle that you guys need to know is it's actually two, one under the right hemisphere and one under the left, that would be right behind this flap of tissue here. That's the lateral ventricles. Okay, so here they are, and they would extend all the way under the cerebrum so that the cerebrum got lots and lots of cerebrospinal fluid. We have another set. This right here would be the space where the third ventricle would be. And then we have what's called the cerebral aqueduct, which would bring it down to the fourth ventricle, which is right here, right where this guy would sit, okay? So, if I can get him in there. Okay, this is the fourth ventricle. Okay. So we have this cerebrum with all its lobes and all that wonderful stuff. The next portion of the brain is called the diencephalon. It's the middle of your brain. So the diencephalon is really this area in here. This is all diencephalon. So the diencephalon is made, first of all, of the thalamus. This is one portion of the thalamus. So notice that the third ventricle, which I said was in here, surrounds the thalamus. So here it is. But the thalamus is really big, and this doesn't give you an indication of how big it is. So I'm going to pull it out and put this bad boy back together again and point out to you that these are the thalami. Okay, this is the thalamus here. Two very large lobes. And running right through here would be the corpus callosum. Okay? Let me put it back where it was, if I can. Aha! I was putting the wrong one in. Okay. So the thalamus, all we can see on the cross section is this part, but we know that it's much bigger than that. It's got these two lobes on it. The other portions are quite small. This area down in here is called the hypothalamus. It's very small. It's this area over in here. It's always going to be closest to the pituitary gland. And then back here in pink, we have the pineal gland. Okay? So this is the what we'll call in the lecture class the epithalamus, which is mostly pineal gland. The pituitary hangs off the hypothalamus by a little stalk. This stalk here is called the infundibulum. I think that's all you guys need to know. Okay, yeah, so now we'll move down to brainstem. The brainstem is made up of the midbrain, the pons, and the medulla oblongata. It contains, as I said, the cerebral aqueduct and the fourth ventricle, which allows cerebrospinal fluid